Okay, so this lecture is on Chapter 2, and what the figure that we have on this first slide is the periodic table of the elements. We're going to come back to this figure in a little bit because it shows us some things like the atomic number, the mass number, about some of the elements that we're going to be talking about. But first, usually what I talk about when I do this chapter is kind of why are we studying chemistry. This is a biology class. Everybody decided to take it because it's a biology class. And so it's a very reasonable question to wonder why we're doing a chapter on chemistry in a biology class. And the answer is that although we're living organisms, we're made up of the same types of chemicals as everything else in our environment. Chemicals compose our body. All body activities are chemical in nature, and that's the case for all living organisms, not just humans. Much of the remaining material in BIO112, and for anyone that goes on to take BIO113, which is the lab, all of that will build on your understanding of the chemical basis of biological systems. If you think back to one of the first figures that we looked at in Chapter 1, which was, I think it was figure 1.2, but it was the levels of organization. And the most basic level of organization of living things is at the chemical or molecular level. So we said at the time that was going to kind of be a roadmap for our semester, and that's definitely the case. So where we're starting is this chemical level of organization. So to do that, we're going to start by talking about atoms. And we're going to talk about the structure of atoms because in a little bit we're going to put together the structure of atoms to become molecules. And by the end of the next chapter we'll have put them together to make really big molecules, what we call macromolecules. And so atoms are composed of three different particles: protons, neutrons, and electrons. Atoms are the building blocks of everything, including human beings and all other living organisms. So let's look at how these particles are arranged in the structure of the atom. There's an area toward the center of the atom that's called the nucleus. And so they show you the nucleus here in the center. The protons and the neutrons are contained in the nucleus. And then the electrons kind of circulate around the nucleus in this area called the electron cloud. We can represent that area by using a line, and we call this an energy shell or an electron shell. Now the other thing that's shown to you here is that protons have a little bit of a positive charge. Electrons have a negative charge. Neutrons don't really have a charge. They're neutral. So in the center of the atom is an area called the nucleus. It's made up of two different types of particles. Protons are positively charged. Neutrons are neutral. Circulating around the nucleus is a type of particle called electrons. And they're found in a region around the nucleus represented by electron shells. All atoms have these parts to them. The way each atom differs is in the number of each of the parts. So they have a different number of protons in their nucleus. Hydrogen has 1 proton. Carbon has 6. Oxygen has 8. Nitrogen has 7. So each atom has an atomic number, or an atom number. We number them, 1, 2, 3, 4, 5, all the way up. And that atomic number corresponds to the number of protons in the nucleus. The number of protons in the nucleus of the atom tells you which atom it is. So wherever you go in the universe, if you find an atom with 6 protons in its nucleus, you know it's carbon. If you find an atom and call it carbon, it should have 6 protons in its nucleus. And then the other piece of information to give is that an atom is electrically neutral. This means the number of positive charges has to equal the number of negative charges. Overall, the pluses and minuses balance out. So, if this atomic number tells you the number of protons, in an atom, it also tells you the number of electrons. So this is a figure from your book that your book is showing you. Hydrogen has an atomic number of 1, so it has 1 proton in the nucleus. It has 1 electron that circulates in this electron shell. Now the other thing that they show you here is that the number of electrons that can go in each shell. The first electron shell can hold 2 electrons. The next electron shell can hold up to 8 electrons. So in addition to showing you the electrons, they're showing you the empty spaces in the outer electron shell. Carbon has an atomic number of 6, so it has 6 protons in the nucleus. It will have 6 electrons circulating around the nucleus. 2 can go in the first electron shell, and the remaining 4 go in the next electron shell. And then we end up with 4 empty spaces in the outer electron shell. Nitrogen has an atomic number of 7. So it has 7 protons in the nucleus, 7 electrons, 2 in the first electron shell, and the remaining 5 go in the next one. That leaves 3 empty spaces. Oxygen has an atomic number of 8. 8 protons, 8 electrons. 2 can go in the first electron shell. The remaining 6 go in the next electron shell. And since that electron shell can hold up to 8, then there are 2 empty spaces. Now the atomic number tells you the number of protons. The fact that atoms are electrically neutral allows us to deduce the number of electrons. But this doesn't tell us anything about the number of neutrons in the nucleus. And neutrons are neutral. They don't really have a charge. So we can't figure out how many based on trying to balance a certain number of charges, because they don't really have a charge to begin with. In that case, what we need to look at is the mass number. There are atoms that have the same number of protons and electrons, but a different number of neutrons. And when that happens, these different versions of the same atom are called isotopes. We identify isotopes by their mass number. The mass number gives the mass of the nucleus. So it tells you all the particles in the nucleus. And isotopes are usually named based on the mass number. So for isotopes of carbon, we have one called carbon-12. It has 6 protons and 6 electrons. Carbon-12 has a mass number of 12. That means there's 12 particles in the nucleus. 6 of them are protons, so the rest are neutrons. Carbon-13 has a mass number of 13. 13 particles in the nucleus. 6 of them are protons, so the rest are neutrons. Carbon-14 has a mass number of 14. 14 particles in the nucleus. 6 of them are protons, so the rest are neutrons. We know the number of protons from the atomic number. Knowing the atomic number with the mass number can help us figure out protons and neutrons. Hydrogen is the same way. Hydrogen's atomic number is 1. So all the versions of hydrogen have 1 proton in their nucleus because hydrogen's atomic number is 1. Each of these atoms has 1 proton. They're all still hydrogen. The way they vary is in the number of neutrons. Hydrogen 2 will have 2 particles in the nucleus. One is a proton, the rest is neutrons. Hydrogen 3 has 3 particles in the nucleus. One is a proton, the rest are neutrons. So these are all isotopes of hydrogen. What varies is the number of neutrons in the nucleus. Sometimes isotopes can be radioactive, but not all isotopes are radioisotopes. So occasionally those extra neutrons are given off or released by the nucleus as radioactive decay. But just because something is an isotope does not necessarily mean that it's a radioactive isotope. So we've talked about protons. We've talked about neutrons and isotopes. Let's talk about electrons a little bit more and these electron shells. So we've said that electrons occupy a series of energy levels or electron shells that circulate around the nucleus. The innermost shell can hold 2 electrons. The next shell can hold up to 8. And the third shell can hold up to 8. That's about as far as we need to go in biology. There are many, many more energy levels and orbitals and shells. But for the purposes of biology, all we need to do is really look at the first 3 electron shells. Another piece of information to think about is that atoms are most stable when their outer electron shell is full. So this outer electron shell determines the reactivity of the element. What chemical reactions it'll undergo. What can it interact with? What chemical bonds can it form? Now, things that are like charges repel each other. Things that have the same charge repel each other. So electrons will repel each other. Opposite charges are attracted toward each other. And that's going to become important as we start to go through and talk a little bit about our chemical bonds and how our chemical bonds are going to form. So this is an additional figure. This is a figure from a different textbook, but I think that it's helpful to kind of summarize what we've been talking about the structure of the atom. So hydrogen has an atomic number of 1. It has 1 proton in the nucleus. It has 1 electron, and that goes in this first shell. The first shell can hold up to 2 electrons. Helium has an atomic number of 2. It has 2 protons and 2 electrons, so its outer electron shell is full. Helium is a very stable element. It's not very reactive. It's so stable, it's so safe that we put it in balloons for kid birthday parties. The second shell can hold up to 8 electrons. So carbon has an atomic number of 6. It has 6 protons in the nucleus. It has 6 electrons. 2 go in the first electron shell. The rest go in the second electron shell. Neon has an atomic number of 10. So its outer electron shell is full. And it's also very stable. Everything that's here on this right-hand side, this actually corresponds to the last column of the periodic table of elements. Those are called noble gases or inert gases, they don't like to react very well. They're pretty stable because their outer electron shell is full. So when atoms want to have a full outer electron shell, for their outer electron shell to be full, they're trying to seek to have a complete outer electron shell. And so what ends up happening is they can be joined together to make molecules held together by chemical bonds, trying to do this, trying to be stable and have a full outer electron shell. There are 3 types of chemical bonds that we'll talk about, and I have them listed here from the strongest to the weakest. So the first kind of bond that we're going to talk about is the ionic bond. And ionic bonds form between ions. So the first thing that we have to do is we have to learn a little bit about ions. Ions are atoms that have gained or lost an electron. So here we can talk about drawing a sodium atom. Atomic number of sodium is 11. So we have 11 protons in the nucleus of a sodium atom. A sodium atom is going to be electrically neutral, so it will have 11 electrons. 2 go in the first electron shell. 8 can go in the second electron shell. And the last one, the 11th one, goes all by itself in the third or outer electron shell. Sodium is not very stable. Sodium is extremely reactive in its elemental or atomic form. If you get on YouTube and you look up sodium atoms or elemental sodium, you can see where lots of people have tried to react sodium with water, and it gives off a lot of light energy and heat energy. So sodium atom is not very stable. It has lots of spaces in its outer electron shell. Now let's draw a chlorine atom. 17 protons, 17 electrons. 2 go in the first electron shell. 8 go in the second electron shell. The rest go in the third electron shell. And that leaves us with one empty space. Chlorine just needs one more electron in order for it to be stable. Atoms like to have a full outer electron shell. Atoms can gain or lose electrons to complete their outer electron shell. So what needs to happen to chlorine in order for it to complete its outer electron shell? It only needs one more electron. What needs to happen to sodium for it to have a completed outer electron shell? You know, wouldn't it be nice if we could just take off this one electron from this outer electron shell and just erase this whole shell? Then there would be a full or completed electron shell. That's exactly what sodium does. Sodium gives up this electron, and that leaves it with a completed outer electron shell. it has lost an electron. Now nothing happens to the nucleus. It still has an atomic number of 11. It's still sodium. Still has 11 protons. But now it only has 10 electrons. It has one extra positive charge. So we draw Na with a plus. It has become a cation, a positively charged ion. That extra electron can jump over to chlorine. Now chlorine has a completed outer electron shell and is also very happy. Still chlorine, still has an atomic number of 17, still has 17 protons in the nucleus. Now it has an extra electron, it has an extra negative charge. So we draw Cl and put a little minus sign behind it to show it's become a negative ion or an anion. Then we have this opposite attract principle. And so this positively charged ion and this negatively charged ion will be attracted to each other. And that is the ionic bond. So ionic bonds form between ions. Ions either have gained or lost an electron to become charged in some way. And then we have this principle in our universe, opposites attract. And so this opposites attract idea gives us this interaction that is the ionic bond. Now another way that we can form bonds is by sharing of electrons. Sharing of electrons creates a covalent bond. What happens if you look at the structure of oxygen, it has two empty spaces in its outer electron shell. Each hydrogen has one empty space in its outer electron shell. And so they can share that pair of electrons and form these covalent bonds. Now in this case, oxygen is an atom that has a really interesting property. Oxygen is what we call electronegative. Look at that up a little more. Oxygen being electronegative means that it holds the electron a little more tightly than other elements do. Oxygen and nitrogen are both very electronegative. So when they share electrons, they don't really share them equally. Electronegative elements hold on to the electrons more tightly. And so this oxygen, being electronegative, will hold on to the electrons a little more tightly. It draws the electrons into it. It gives this area of the molecule just a little bit of a negative charge, slightly negative charge. And it gives these hydrogens a slightly positive charge. That's just saying that the tendency is for the electrons to kind of stick closer to this oxygen molecule. So the sharing of electrons in oxygen is unequal. We have this unequal sharing, and we call that a polar covalent bond. We have unequal sharing of the electrons because oxygen is electronegative, and that creates a polar covalent bond. So these polar covalent bonds occur when hydrogen creates covalent bonds with other elements that are electronegative, like oxygen and nitrogen. In a polar covalent bond, the electrons are not shared equally. The electrons stay closer to the electronegative atom. So we have unequal sharing of electrons to create this polar covalent bond. That sets up the conditions for our third kind of bond, which are hydrogen bonds. Hydrogen bonds form because of polar covalent bonds. So molecules containing polar covalent bonds have an area with more electrons that has a little bit of a negative charge. And it has an area that has a little bit of a positive charge. So the partial positive charges from adjacent molecules are attracted to the partial negative charges. This opposite attract principle, and that is what creates the hydrogen bond. So we have this opposites attract principle. We have polar covalent bonds that make a little bit of a positive charge on hydrogen and a little bit of a negative charge on the oxygen. And this forms an attraction between two water molecules. Here's an image that shows that. It shows this dotted line as the hydrogen bond. And it's an attraction between the negative on one molecule and the positive on another molecule. You have this slight positive charge and this slight negative charge, and there's an attraction there. That attraction is called the hydrogen bond. And we're going to have that anywhere we have two molecules that have a polar covalent bond. That's going to be anywhere where you have oxygen or nitrogen bound to hydrogen. We're going to see that when we go through structures of proteins. We're going to see that when we go through structures of carbohydrates. So where we have these polar covalent bonds, we can form hydrogen bonds. So we have three kinds of bonds. The first one we talked about is the ionic bond. It occurs between ions. Loss or gain of electrons creates an ion. And then you have this opposites attract idea. Then we talked about covalent bonds, which is sharing of electrons. The goal is to complete the outer electron shell of both atoms. Because we're sharing electrons, this is the strongest of the bonds. And it can either be equal sharing, it's a non-polar, regular covalent bond, equal sharing of electrons. Or if you have unequal sharing, that's a polar covalent bond. The unequal sharing might be because of electronegativity. That sets up the conditions for hydrogen bonds. Hydrogen bonds are an opposites attract association between adjacent molecules that have polar covalent bonds inside of them. And then here's a table that kind of summarizes these three types of bonds. So we started out by talking about the structure of atoms. Atoms contain protons, neutrons, and electrons. And make sure that you understand kind of the configuration of those. Atoms can combine to make molecules by forming chemical bonds. Ions can make ionic bonds. Sharing of electrons can make covalent bonds. And then polar covalent bonds set up the conditions for hydrogen bonds. So we started out by talking about the structure of the atom. That told us where we had empty spaces in the outer electron shell to know where we needed to fill in or move electrons around, tell us what we need to do to get a completed outer electron shell. Because atoms are trying to have a full outer electron shell. And in doing so, they often interact with other atoms. So now we have formed some molecules. Now a lot of times, the forming of chemical bonds occurs in a chemical reaction. So chemical reaction occurs when we combine things to form products, and a lot of times we're combining things to try to have a full outer electron shell. Chemical reactions can be written as chemical equations. So on one side, we start with the reactants, the things that go through the chemical reaction. We draw an arrow to show the reaction has taken place, and at the end of the arrow, we write the product of the reaction. Now, some reactions are facilitated by enzymes, and so we'll write the name of the enzyme above the reaction. Two reactions that we're going to talk about are dehydration synthesis and hydrolysis. And so we're going to talk a lot more about these two reactions in Chapter 3, and then we're going to talk a lot more about enzymes in Chapter 5. So we'll come back and tie those topics in to chemical reactions. So this is a figure that your book shows you to show you this idea that we have reactants. We draw an arrow to show that the reaction has taken place. We have formed some new bonds in creating our product. And in doing that, these atoms are trying to have a completed outer electron shell. They're trying to be as stable as possible. Now, another thing to look at as we're talking about some basic chemistry ideas is we're looking at solutions and some terminology related to solutions. So a solution is just a mixture of two or more substances. And the terms that we use to refer to the substances are solvent, which is what does the dissolving, and the solute, which is what's being dissolved. So the example that I almost always use when I teach these terms about solutions is the concept of how do you make Kool-Aid. So whenever anybody's making Kool-Aid, usually you put your packet that has your flavor and color molecules, a cup of sugar that has sucrose molecules, and then you add water. Water is the solvent, and the color and flavor and sugar molecules are the solutes in your Kool-Aid, which is an aqueous solution. If water is the solvent, then that's an aqueous solution. Now, if the solutes have polar covalent bonds, they tend to dissolve very readily in water. They can form hydrogen bonds and dissolve very readily in water. So we call them hydrophilic. Substances that have non-polar covalent bonds are hydrophobic. These are usually things like long hydrocarbon chains with very equally shared electrons. And so we're going to see a little bit more about how the structure causes hydrophilic and hydrophobic compounds in the next chapter as we start to look at our big macromolecules, carbohydrates and proteins. Now, another topic when we talk about solutions to learn about is pH. pH is a measure of the hydrogen ions that are in solution. pH very literally means potential hydrogen, and so pH is a measure of how much hydrogen ions you have in a solution. All solutions are either acids or bases or neutral. They all fit into one of those categories. The definition of an acid is something that releases hydrogen ions into solution. So if you have something like hydrochloric acid, the chemical formula for hydrochloric acid is HCl. In solution, it becomes a hydrogen ion and a chloride ion, hydrochloric acid. Bases take the hydrogen ion out of solution, usually by releasing something called a hydroxide ion or hydroxyl ion into solution. Because when you combine hydrogen ion and hydroxyl ion, you get two H's and an O. The combination of these is water, which is perfectly neutral. So when we have this hydroxide ion released into solution, it can combine with the hydrogen to form water, which is perfectly neutral. We measure the relative hydrogen ion concentration using the pH scale. The pH scale runs from 0 to 14. Neutral is 7.0, an exactly equal amount of hydrogen ion and hydroxide ion because they form water. Anything less than 7 is acidic, and anything greater than 7 is basic. So let's take a look. This is the figure from your textbook. Again, if your amount of hydroxide ion and hydrogen ion is exactly equal, you're at 7. Pure water is neutral. Anything with a greater number than that, anything up to 7, is basic. And anything that's lower than 7 is acidic. This pH scale is actually based on a logarithmic scale, which means that pH 10 is actually a concentration of 10 to the minus 10 hydrogen. pH 4 is actually 10 to the minus 4 hydrogen. So this is a bigger number than this. I tell you that not to make it seem confusing, but actually to try to clear up something that tends to be confusing. So let's look a little more closely at what is written on the scale. This says lower hydrogen concentration as we increase in number. And this is greater hydrogen concentration as we decrease in number. That seems counterintuitive. It's based on the exponent that I was just showing you. But what you'll need to remember is anything that has a lower number is more acidic. Tomato juice, grapefruit juice, lemon juice, battery acid, stomach acid. As you get a lower number, you get more acidic. As you get a higher number, ammonia, bleach, oven cleaner, you get more basic. Higher number is more basic, lower number is more acidic. So it seems a little strange the way they have it written. The lower hydrogen concentration is the higher number. Really what that's saying is a more basic solution has a higher number, and a more acidic solution has a lower number. And so finding a way to try to keep those separate is going to be the best way to help you kind of understand this idea about the pH scale. Sometimes it's just kind of thinking about it or looking at it, working with it a little bit, but it can seem counterintuitive, and that's usually why I take a few minutes to try to explain to folks what it is that you're looking at here. Because when you first glance at it and you see lower hydrogen concentration as a higher number, that can sometimes be confusing. But that lower hydrogen concentration just means more basic, the more basic solution, less hydrogen. So chapter 2 goes through a lot of things related to atoms. So this periodic table, pretty much the atoms that we're going to be dealing with in human systems are in the first 2 or 3 rows of the periodic table. Usually a periodic table tells you an atomic number. Remember, that corresponds to the number of protons. And it'll tell you a mass number. That's everything in the nucleus, protons plus the neutrons. And what we did was we figured out the arrangement of protons and especially electrons, because the electrons help us understand what kind of bonds we're going to form, what kind of reactions we're going to have. The empty spaces in the outer electron shell give us a lot of clues because atoms are trying to fill up that outer electron shell. They fill up that outer electron shell by forming bonds, and those bonds are going to create molecules. Now in Chapter 3, we're going to start getting into bigger molecules, macromolecules, carbohydrates and proteins. And so if we keep in mind how these atoms are fitting together into molecules, that's going to make part of what we study about the macromolecules in Chapter 3 make a little bit more sense. Okay, so this is going to be on Chapter 3. This kind of is a little bit of a continuation of what we were doing in Chapter 2. In Chapter 2, we started with the atom and we did structure of the atom and talked about how there are empty spaces in the outer electron shell. Hello?
I'm waiting for her.
Okay, so this is Chapter 3, which is going to be kind of an extension of Chapter 2. In Chapter 2, we started with the atom, looked at the structure of the atom, and looked at the spaces in the outer electron shell, because those spaces would tell us what kind of bonds that atom is trying to make, because atoms are trying to complete or fill up their outer electron shell. And so they would form these bonds with other atoms to try to do that. In Chapter 3, we're going to build on that idea of those bonds forming, and we're going to look at making bigger and bigger and bigger molecules from those. So in this chapter, we're talking about organic compounds. Sometimes these are called macromolecules, really big molecules. And there's 4 major classes that we're going to talk about. We're going to talk about carbohydrates, lipids, proteins, and nucleic acids. The basis of all of these is that they have a lot of carbon in them. All of these macromolecules or organic compounds have a lot of carbon bound to hydrogen. So carbon has an atomic number of 6 protons in the nucleus, 6 electrons, 2 go in the first electron shell, and the remainder go in the second electron shell. And since that second electron shell can hold up to 8 electrons, we end up having 4 empty spaces. Carbon can form bonds with 4 different things. And so if we look at it kind of in a structural way, we can denote the bonds with lines. Here, they're denoting each atom with a ball and the stick in between them showing the bond. And this is a space-filling model. If we actually think about that electron cloud idea, then this is a space-filling model. But the basic thing to understand is that carbon can create 4 covalent bonds to other atoms, and those atoms can also create covalent bonds. So we can start making these big chains of macromolecules. So carbon backbones can be short or can be really long. We can have branched chains, because carbon can have all 4 bonds, and one of these bonds can go out and to the side and make another chain. Carbon can form these ring structures. So there's lots of different ways that carbon can be arranged. So carbon is pretty functional for making these long macromolecule chains. In addition to carbon chains, there's another important thing to talk about with molecules, and that's the idea of functional groups. There's 2 common functional groups. One is hydroxyl groups, one is carboxyl groups, another is amino groups. We'll talk about when we get to those amino acids. Now, some things to look at here with these. There's some oxygen bound to hydrogen, so what you should immediately be thinking is electronegative atoms. That makes a polar covalent bond. These are going to make molecules polar, which means they're going to be very soluble in water. These are very hydrophilic kinds of macromolecules if they have these kinds of functional groups. So in Chapter 2, we talked about once you start putting molecules together and creating chemical bonds, that we start doing that by chemical reactions. And there are two important chemical reactions that we study with these macromolecules. One is called dehydration synthesis. It's a synthesis reaction. It's how we build up our long macromolecules. The other is hydrolysis. The lysis or breakdown reaction, and it's how we break down all of our macromolecules. So first they introduce these chemical reactions in a very general way, not really specifically talking about much of the structures at all, except for this part of the structure and this part of the structure, because they're going to join together to form water. You have an H and an OH, and together they're going to form water. When they do that, a bond is going to form to make this longer macromolecule, this longer polymer. Polymers are long chains of repeating subunits. One subunit is often called a monomer, and then lots of them together is a polymer. So when we make these big macromolecules, it can be a long carbohydrate, it can be a protein. It's made up of these repeating subunits. We can also break down a long polymer, a long carbohydrate, or a long protein. When we add water, water is going to break this bond. Part of the water molecule ends up as the OH, part of the water molecule ends up as the H. And so we've broken that bond and broken down that polymer. These reactions are the exact opposite of one another. We can form water, that's the dehydration synthesis, to make a longer polymer, or if we add water, we can break a longer polymer into its subunits, and that's hydrolysis. These are exactly the opposite reactions. These are exactly reversible reactions. And so as we go through and look at each of our macromolecules, you'll see that each one can be built up and broken down using these two reactions. Now we can summarize our polymers and monomers in a chart. So on one side we can put our macromolecules. These are our polymers. And on the other side we can put the subunits that they're made of. These are the monomers that these are made of. We've got four main macromolecules that we're talking about. We're talking about carbohydrates. We're talking about lipids. We're talking about proteins. And nucleic acids. Carbohydrates are made up of single sugar subunits, things like glucose, fructose, galactose. Lipids are made up of fatty acids, and there's a whole bunch of them. We're not going to really learn many names of fatty acids. Proteins are made up of amino acids. There are about 20 amino acids that make up proteins. And nucleic acids are made up of something called nucleotides. And there are 4 nucleotides that make DNA and 4 nucleotides that make RNA. The DNA nucleotides are A, C, T, G. The RNA nucleotides are A, C, U, and G. All of these get built up by dehydration synthesis. All of these get broken down by hydrolysis. Those two reactions that we just looked at put together all of these, put together DNA, put together proteins, put together lipids, put together carbohydrates. In the same reaction, hydrolysis breaks down carbohydrates, breaks down lipids, breaks down proteins, and breaks down nucleic acids. So let's start to look at these one by one. Carbohydrates is the first of our macromolecules to talk about. It's made of carbon and water, so that's where the word carbohydrate comes from. It's a really important energy source for metabolism. We're going to take electrons off of carbohydrates and put them through something called the electron transport chain to make cellular energy. And we're going to talk about how that happens in a few weeks. Single sugar subunit is often called a monosaccharide. Two sugar subunits together is a disaccharide. And then if we form a bunch of them, that's a polysaccharide. And we form the disaccharides and the polysaccharides by dehydration synthesis. So the way that they show the sugar subunits most of the time is this kind of ring structure. So we're going to string a bunch of these little ring structures together as we go through. You're not required to learn a lot of structures, so you're not going to need to learn either the linear or the ring structures. But when you look at some of these long chains, what you'll need to do is recognize that each one of these is a sugar subunit. So, for example, in this long, not very long, disaccharide of lactose is made up of these two sugar subunits. So we have an OH on one of them, an H on one of them, and that forms water. This is dehydration synthesis. That's forming a new bond between them, and that's making a disaccharide called lactose. We can make even longer carbohydrates. Polysaccharides can be very long. They can be hundreds of sugar subunits long. One that's important is called glycogen. This is how animals store extra carbohydrates in the liver or the skeletal muscles. And so when blood sugar drops, the liver hydrolyzes the glycogen to yield individual glucose subunits. And so here they're showing you glycogen in muscle tissue or in the liver, which are these long carbohydrate chains. And so they're showing you each one of these is a glucose monomer. And so starch is a long chain of those. Glycogen is a long chain of those. Plants have cellulose, which are these long chains. Now our next macromolecule to talk about are lipids. And like carbohydrates, lipids have carbon and hydrogen, but they don't have as much oxygen. Have a little bit, but not as much. So they have very few polar covalent bonds. Lipids are more long hydrocarbon chains with very equal sharing of electrons, very nonpolar covalent bonds. So they're very hydrophobic, and they're not very soluble in water. So this is supposed to be figure 311 from your book. Figure 311 in your book shows some long long fatty acid chains. These hydrocarbon chains can be very long. It shows the H and the OH, and so it shows how we build up this triglyceride by dehydration synthesis, the exact same molecule. But you can see how there's very little oxygen, especially in these fatty acid tails. So it makes them very, very hydrophobic. And then other lipids are things like cholesterol. Cholesterol can be converted to other compounds in the body that are also very lipid soluble or fat soluble, that are not very soluble in some body fluids that are aqueous solutions. Now we're going to talk more about lipids in the next chapter when we talk about phospholipids, because they create the structure of the cell membrane. So phospholipids are going to be really important when we talk about cell membrane structure and function. Our third macromolecule is proteins, and proteins are very important to body function. They're constructed from about 20 amino acids. We're going to talk here in just a second about the structure of amino acids. Then we're going to talk about how they're put together to form a protein. We're going to need to talk about how proteins fold. This is the first of our macromolecules that have to be folded in a particular three-dimensional structure in order to have their individual function. When we look at the amino acids, the general structure of the amino acid is going to be the same. What's going to differ is a part called the side chain. These side chains contain different functional groups. That's going to help with protein folding. One thing that's unique here in the amino acids is the bonds that we form by dehydration synthesis has a special name called a peptide bond. So if we form two amino acids, that's a dipeptide. If we form longer amino acid chains, that's a polypeptide. The basic structure of an amino acid forms around a central carbon. And carbon can bond to four things. So on one side is an amino group. It has a nitrogen down to some hydrogen. On the other side is a carboxyl group. There's lots of oxygen here. The third thing is just a plain hydrogen. And then the part that varies among different amino acids is the side group. So the amino group, the carboxyl group, and the hydrogen around the central carbon are the same for all amino acids. What differs is this side group. It can be very hydrophobic. This is all just hydrocarbons. There's no oxygen or nitrogen here. Or it can be hydrophilic. There's some oxygen here to make this side chain polar. There's 20 different amino acids. Some have long side chains, some have short side chains, some have different functional groups that can create covalent bonds. And so we have an H and an OH, so we can form dehydration synthesis and create a new bond. And again, in a peptide, it has a special name, the peptide bond. And we can continue to add and add and add amino acids and string amino acids together to make these long chains. So proteins have to be folded properly in order to function. So we can make these long chains of amino acids, but to actually form a protein, it has to be folded into a three-dimensional structure. The first step is forming peptide bonds to get this long chain of amino acids. But then we get interaction between side chains, between those, between different amino acids, to kind of fold up the protein into a specific three-dimensional shape. That three-dimensional shape is usually held in place by hydrogen bonds or maybe covalent bonds. So the first level of protein folding, primary protein folding, is the amino acid sequence. Here they're showing you 129 amino acids all strung together. They're giving you the name of each amino acid kind of in a string. So this is the first step to making a protein. But this protein will not be functional until it has some sort of folded or three-dimensional structure. So once we have this sequence of amino acids, the primary structure, then we have the second step, secondary structure, where we make these either pleated sheets or an alpha helix, and these are stabilized or held in place by hydrogen bonds. We then fold the whole protein into its three-dimensional shape. That's its tertiary structure. Sometimes those polypeptides associate with other polypeptides to form a protein that has subunits. For example, hemoglobin is a protein that has subunits. Those proteins have quaternary structure. There's a video that's posted in Blackboard. In the Learning Materials tab, there's a folder called Protein Folding and Enzymes, and so it has some videos. It has a really good YouTube video. It's very short, but it's put together by a lab that studies protein folding. It doesn't have any sound to it, but what they show you is they show you a string of amino acids very similar to this, and they show you how it folds, how it forms an alpha helix, how all that wraps up together, and how that forms the three-dimensional functional structure of that protein. The protein folding or shape or conformation determines its function. If it's not folded properly, it can't function properly. And proteins that are pushed outside their optimal range of different conditions can become unfolded or unraveled. And that's called denatured or denaturation. Temperature, salt, pH, all can denature or unfold proteins. Here's an image from your book that shows you a protein, kind of a space-filling model of a protein, and it gives you the idea that there's some areas of this protein that have to have a specific structure in order to fit their specific target or to do their specific function. In a couple chapters, we're going to talk about enzymes. Enzymes have an area called the active site that binds to the substrate for the reaction that they're catalyzing. And that active site has to be folded properly in order for that substrate to fit. Other misfolded proteins are associated with diseases. So here is a segment of a protein for normal hemoglobin, and a mutation has changed just a single amino acid, and now this protein will not properly fold and cannot help the red blood cell keep its normal shape. The amino acid sequence is determined by the DNA. We're going to go through a couple of chapters where we talk about DNA. We're going to talk about how DNA determines the amino acid sequence of a protein. And if the DNA changes, the amino acid sequence changes. So a mutation in the DNA can lead to a misfolded protein, and that can cause a disease process. So our fourth macromolecule is nucleic acids. The function of nucleic acids is to store information and to kind of direct some cell functions. Specifically, nucleic acids like DNA are going to direct the synthesis of proteins, just like we talked about in that previous slide. So you have a gene in the DNA. DNA is a double-stranded or double helix molecule. DNA stays in the nucleus of the cell. DNA doesn't get out of the nucleus of the cell. If the DNA ever gets out of the nucleus of the cell will kill itself. Proteins are made on ribosomes that are in the cytoplasm of the cell. We're going to go through the parts of the cell in a chapter that we're going to have next week. So in order for the information that's in the DNA to get to where the protein is made on the ribosome, you have to have a messenger molecule go between them, and that's what RNA does. RNA carries the information that's in the DNA to the ribosome so that amino acids can get strung together in the correct order to make a protein. Nucleotides are composed of a sugar, a phosphate, and a base. And it's the base that we talk about when we usually talk about nucleotides. We usually abbreviate them by their first letter. A, T, C, and G are the DNA bases. A, U, C, and G are the RNA bases. There's no T in RNA. Instead, it's uracil. And when we get into talking about DNA structure in a few chapters, we'll talk about the details of what DNA bases do. But essentially for now, what we can do is look at a simplified, so we have this huge structure that is this nucleotide. And again, you don't need to memorize specific structures. They simplify it down to a symbol here, where the yellow and blue are going to form the backbone, and the base is going to stick out to the center. Eventually, it's going to form the double helix that we're familiar with DNA. So we have this long backbone with the bases that stick out. And we put two of these strands together, wind it around, to form the DNA double helix. And the way the DNA double helix stays together is by hydrogen bonds. So the hydrogen bonds that we talked about in Chapter 2, hydrogen bonds that can occur between molecules that have polar covalent bonds. If you go back and look at the structure of the base, you're going to see lots of oxygen. So there's lots of polar covalent bonds. Lots of opportunity for those bases to form hydrogen bonds. So in this chapter, we've been talking about our macromolecules. We have 4 macromolecules: carbohydrates, lipids, proteins, and nucleic acids. They're all polymers, which are long chains of repeating subunits. The subunits are all put together by dehydration synthesis and broken down by hydrolysis. And we can have folding or other structures occur because of hydrogen bonds. So protein folding, the alpha helix, occurs because of hydrogen bonds. DNA double helix occurs because of hydrogen bonds. Each of our macromolecules are made up of subunits, and those subunits repeat. And we put them all together by the same reaction, and we break them down all by the same reaction.